Okay, this week we're talking about seeds. Here are just some different seeds, just to show you diversity of what they look like under magnification. So a seed is the unit of reproduction. It's also a miniature plant in a arrested state of development. So within a mature seed, you're going to find an embryo, a seed coat, food storage, which would have the endosperm, and cotyledon, which is the seed leaf. Okay, so here's a picture of a bean on the left, which is a dicotyledon, and a corn seed on the right, which is monocotyledon. And just to go over some of the terminology you see here, your book will also cover this. The epicotyl will become the shoot, stems, and leaves. The cotyledon is the seed leaf. The radicle is the embryonic root. The hypocotyl is the portion between the cotyledon attachment and the radicle. It helps elevate the cotyledons above the soil surface. And the endosperm is where food reserves are stored. So monocotyledons, or monocots, remember that's a subclass when we're talking about our classification last week. They have one seed leaf, and these contain grasses, lilies, daffodils. We can see tulips here, bamboo, palms, agave. And then with two cotyledons, or seed leaves, dicotyledons, and this is everything else that has true flowering or fruits. So the big differences here are the monocot has a single seed leaf or cotyledon. The dicot has two. Monocots tend to have long, narrow leaves, and their veins tend to be parallel. Dicots tend to have broad leaves, and they have netted veins. Within the stems, you will see the vascular bundles that are scattered within a monocot, and we'll talk about this soon. The vascular bundles in a ring on dicot, and then with the flowers, the floral parts are going to be multiples of 3 for a monocot and 4 to 5 for a dicot. Okay, so here's another graphic and talking about the roots, and for the most part, monocots are going to have fibrous roots. A lot of dicotyledons do have tap roots, but they also have fibrous roots, so don't let that fool you. You can have dicotyledons with fibrous roots. Just be aware of that, and that's going to be like annuals. Again, our parts are hypocotyl, the part between the roots and the cotyledons. The epicotyl, which is where an embryo or seedling stem above the cotyledon. And then the radicle is the embryonic root. Okay, so viability is when a seed is capable of germinating after all the necessary environmental conditions are met. The average lifespan of a seed is 10 to 15 years if everything is correct in storage, and this would be cool and dry. That's what seed banks maintain. Some plants require light, others don't. So rule of thumb, if a seed is really small, it's most likely going to require light to germinate. So you're going to need to barely cover those seeds. They all require moisture. Temperatures can vary widely, and they all need oxygen. And think about soils that are frozen, compacted, or waterlogged. They're not going to have much in the way of oxygen, so seed germination is difficult in those situations. And I just wanted to go over the fact that river birch seeds are non-dormant if they're germinated under long days and where they get light for 16 hours straight. But if they don't get that, they do need to be stratified for 60 days. And I'll talk about stratification as a little bit later in this presentation. So water uptake softens the inner tissues. It causes swelling and the seed coat to rupture. It also allows for more water uptake. And just so you know, the alurone cells is the outermost portion of the endosperm. So seed dormancy prevents germination under poor conditions caused by hard seed coat, immature embryo, and chemical inhibitors in seed and fruit. And the most common inhibitor is abscisic acid, which is ABA, and plants such as sugar maple, red oak, and English oak are among the species that contain ABA in their seeds. Allelopathy is something that happens when a plant releases chemicals to negatively impact another, so seeds may not germinate if they're under one of these plants. And the classic plant that is allelopathic is the black walnut, not the English walnut, but the black walnut. And it's different from competition. Competition is where resources are scarce and only so many plants can get to it. This is a chemical way to prevent other plants from growing near their plants. So overcoming dormancy for annual plants, temperature is most important, but with other plants, time is important, so you can have the embryo mature. You might need to have warm, cold, moist conditions. Stratification is when you have cold and moist conditions, and scarification is when you're breaking a seed coat. And you can see here they're using a file to break the seed coat, so that's scarification. Stratification usually takes 60 to 90 days. It's best at 35 to 40 degrees Fahrenheit and kept moist, and you do want to make sure you're preventing critters from getting to them. Scarification is when you artificially break down the seed coat. You can do this mechanically. Here where they're using some sort of blade. You saw that file previously. You can use hot water, allow it to soak overnight. You want to sow it soon so you can keep it moist. You can use hydrochloric acid. And then some plants need heat, and in this case, naturally occurring lupin is scarified during a fire, and that stimulates rapid germination. So germination occurs as the result of the swelling of the endosperm, the rupture of the seed coat by the emerging root, and the emergence of the shoot.